All right, let's move on to the chromosomal theory of inheritance. Now, the chromosomal theory of inheritance, actually, the idea that something inside of the cell gave rise to the passage of traits from parents to offspring, that idea had been around for a while, even before Mendel's studies. A few other people, other researchers, had noted that, but Bovary and Sutton really came up with this idea that, hey, maybe something about the way chromosomes look during mitosis has something to do with how traits are segregated and distributed to offspring during fertilization. And so the chromosome theory of inheritance states that the inheritance patterns of traits can be explained by the transmission of chromosomes during meiosis and fertilization. So remember, when we looked at meiosis, we had a couple different things going on there. We had mom and dad's sister chromatids coming together, and then crossing over might occur sometimes. And we also had mom and dad's sister chromatids aligning independently along the metaphase plate during meiosis 1. So not every time are you going to get all of dad's chromosomes on one side and all of mom's on the other side. Sometimes you're going to have a flip, right? And so that leads to gametes having more or less of mom and dad's chromosomes in the nucleus. So basically, you have a mixing of traits, a mixing of chromosomal composition during prophase 1 and metaphase 1. And so here are the observations that gave Bovary and Sutton and a few other people an idea that maybe the chromosomes had something to do with distributing genetic information. So chromosomes are made of genetic material, nucleic acids. Chromosomes were replicated before they were passed to the offspring, before meiosis occurred. Another thing was that the nuclei of most eukaryotic cells have chromosomes that are homologous. In other words, one chromosome, one from mom, and one chromosome, one from dad. And so that gives you an idea that, hey, both mom and dad have put information into that cell, and it's how that information is expressed that develops into a trait. We do know that that diploid number gets reduced to the haploid number, and that haploid number generates independently distributed contributions between mom and dad into the different gametes. And each parent contributes one set of chromosomes. So here is what we were just talking about. During meiosis 1 prophase, you have moms and dads sister chromatid pairs. They align next to each other, and if no crossing over occurs, mom and dad's chromatid pairs separate during anaphase and telophase 1, which ends with cytokinesis 1, and that is our first reduction division. So we went from diploid here in prophase 1 to haploid at the end of cytokinesis 1. And then the cells just go through division again. There's no more going through G1, S, and G2. The cell just divides the sister chromatids into individual chromosomes. Heads up, guys. Whenever you see sister chromatid shapes, the cell cannot survive like that and do metabolism. These really tightly condensed chromatid pairs don't completely silence genes, but the cell can't make a whole lot of new protein when chromosomes are really tightly condensed like this. So for the most part, you're only going to see gametes maybe hanging out in this particular configuration, chromosomal configuration, in pretty much, most somatic cells. You're going to see looser combinations or looser organizations of chromosomes, and they're not going to be sister chromatids. They're going to be individual stick shapes. So X's only occur when the cell's getting ready to divide. Now, this is what we were talking about in terms of independent assortment. So here's like chromosome 1 of mom, chromosome 1 of dad, chromosome 2 of mom, chromosome 2 of dad, and you're looking at color, yellow, green, round, wrinkled. Remember the yellow and green and round and wrinkled peas? If meiosis occurs such that all of dad's chromatid pairs align on the left and mom's on the right, then you'll end up getting two gametes that are exclusively looking like mom and two gametes that look like dad. Over here, though, in this particular setup, mom and dad's chromosomal compositions have mixed. Dad's is here, mom's is here, and then mom and dad are aligned oppositely. So when the cell goes through meiosis 1 and sister chromatid pairs are separated, the chromosome composition is mixed, 50-50, half mom, half dad, so that ultimately in the gametes, you have what we call recombinant gametes. These are new combinations of chromosomal composition compared to what you originally saw in the parental cell. So this is really interesting. This is how you can have mom's and dad's information either maintained separately by chance or more likely mixed up. So what does this mean in terms of humans? So it's really difficult to study humans genetically because humans, unlike garden peas, have very long generation times. Pregnancy is 9 months long. It takes up until 18 to 20 some odd years before the individual's before the human is an adult. So some traits you may not see quickly. Usually what geneticists do to study human traits is do a pedigree analysis. On top of, simply, generation time, it's pretty unethical to plan hybridization experiments with humans. So basically what a pedigree analysis is a family tree. And family trees or pedigrees are often represented this way. So females are circles, males are squares. An affected individual for a given trait would be a solid colored in shape. An unaffected would be a blank shape. A deceased individual has a line through it, and so forth. You can go on ahead and read through the key here. In this particular family tree, what you're seeing is the male and a female that are presumed heterozygotes because you have one affected male here, a presumed heterozygote female, a presumed heterozygote male, and then when you look at this female who married this male, and you look at the offspring, you'll see which ones are affected and which are unaffected. What this implies is that you would have a homozygous, homozygous, heterozygous, heterozygous. These two would be homozygous as well, just unaffected, and this would be homozygous affected. So a pedigree analysis is used usually by genetic counselors to help determine the pattern of inheritance of given diseases and given traits in families, in human families. Although pedigree analysis is often done for dogs, cats, agricultural animals, and plants as well. Usually what you're looking at is a normal allele, and then you will be looking at a mutant allele that gives you the disease symptoms or that gives you the traits that you're interested in looking at. And oftentimes you're hoping that the Mendelian pattern of inheritance is dominant and recessive because then it's real easy to track the trait. So, what do you need to know for the exam? There are specific patterns of transmission for traits that are recessive or dominant. A recessive pattern of inheritance, 25% of the individuals will end up showing the effect. If you think of the Punnett Square, homozygous dominant and the two heterozygous individuals are not going to show the trait because you have to have two recessive alleles to make the trait work out. So in other words, this is what we're looking at here. Let me draw this out. I need to figure out how to get my pen working. So let's say over here, let's see if this will work. Okay, not working. I'm going to have to figure this out. My apologies. Let's come back to this Punnett Square. You should see how it is in class. Sometimes I have to go back all the time and look at individual Punnett squares. So let's go back to our monohybrid cross. Right here, when a trait is transmitted recessively, this is the only individual that will show the disease. Each one of these quadrants has a dominant trait or a dominant allele, which is a rescuing allele. So if you don't have the dominant big T, your phenotype is not rescued, and therefore, if you have little t, you have that particular disease. So let's come back over here to our recessive disorder. So an unaffected heterozygote individual will average 25% of their offspring being affected. So again, go back to drawing that heterozygous monohybrid cross, and you'll see that that's, the little t would be the affected. Two affected individuals, okay, are going to produce 100% offspring that are affected. So if you have two individuals that are little t crossed by little t, they're all going to be little t. Interestingly, you can see a skipping of generations in the recessive pattern of inheritance. In other words, you can have a situation where you have big T, little t, and everyone basically does not show the disease, but in the next generation they might. In a dominant pattern of inheritance, the disease does not skip generations. The disease is obvious in all generations, and that's because big T is what gives you the disease. So an example of a recessive disorder, cystic fibrosis. Cystic fibrosis is a pretty nasty disease where you have this receptor or regulator. It's a chloride channel regulator called a transmembrane conductance regulator. It controls the amount of chloride that goes across the cell membrane in cells and facilitates the production of mucus. And so what ends up happening is that you have an ion imbalance that leads to a buildup of very, very sticky mucus in like the intestine, in the lungs, definitely, sweat glands, reproductive organs, and that leads to lots of usually infections and early death in individuals. And so in this particular case, you'll see two parents that are heterozygous for the disease, but they're not showing it. They don't have it. In this case, this individual is the affected offspring. This individual, they're assuming, is heterozygous for the disease, even though they're not showing that they have it, right? Only the ones that are filled in all the way are the affected individuals. We assume that this individual and her mate are also heterozygous because they have two offspring that show cystic fibrosis. So the last video that I'm going to do is the probability and statistics video. What I need you guys to understand about probability and statistics is that basically a lot of what's going on in genetics is kind of like Vegas, all right? Animal breeders, genetic counselors all use simple statistics to predict whether or not individuals are going to demonstrate specific traits. So you need to know the genotype of the parents, or at least you need to have an idea of what the genotype is, so that you can predict the phenotype of the offspring with really good reliability. Sometimes it can be very difficult to do that, though, depending on the level of expression you're looking at. Are you looking at the whole individual, or are you looking at just individual cells in the individual? So what I want you guys to do for probability is to definitely review the math in terms of flipping coins. We all know that if you flip a coin, you have a 50% chance of getting a heads or tail. What probability is the possibility that an event is going to occur in the future. You can calculate that by the number of times the event occurs divided by the total number of possible events. So if you flip a coin and you're looking for the probability of heads, you only have one head on the coin, and there are two possible outcomes, head or tails, so you add those together. That reduces to 1/2, which is 50%. So the odds of you flipping a coin and getting heads is 50%, which is pretty easy. Now, when you're doing this type of analysis, I mean, that's a really simple example. When you're doing this type of analysis in genetics problems, it's difficult to do that with very few or very small sample number. Usually, experiments are going to have a real large sample size so that you can perform the experiment numerous times so that you can say, okay, I feel that I have a good confidence that the outcome will be 70%, let's say, given this particular example. So in random sampling is going to be looking at a random number of individuals, or you set a number of individuals, but you're going to sample just a few within that larger set to figure out whether or not your assumption is correct. We'll move on with this with basic statistics in our last video.